नमस्कार दोस्तों, वेलकम टू चैनल टॉक्सिकोलॉजी ऑन बोर्ड. In continuation with ICH-S9 question and answer document, today we'll discuss part 2. ICH-S9 is is there in the part of ABT brochure 2026. So let's directly dive into the question. So these are the questions from the Q&A document before the guideline came into existence. So these are the questions from the scientists from the pharmaceutical industry. So part 2 will be the questions which were related to to studies to support non-clinical evaluation. So the question number 8 is in section 2.1. 2.1 discusses about the pharmacology. The guidance states that means ICH-S9 guidelines states that the study should characterize the anti-tumor activity of the pharmaceuticals. Whatever the anti-cancer drug which you are developing for the treatment of patients who are in advanced cancer stage, this particular drug, you have to establish anti-tumor activity of this particular pharmaceutical investigational new drug. The inference is that these are in vivo studies because how we are going to establish the efficacy? Cancer cell lines you're taking, you're putting your drug, and you're checking, or in the case of tumor models also, the inference is that these are in vivo studies. Is in vivo characterization necessary to address this pharmacology? This is a question from the scientist. The answer which is given by the regulatory agency is if in vitro systems are used for pharmacological studies, in vitro means cell lines of anti-tumor activity are demonstrated to generate relevant data, then they should be considered sufficient. So what is here the agency is going to tell, ICH regulatory body, is that if you have an in vitro data in which this particular IND drug has shown, anti-cancer drug has shown efficacy, it was able to establish anti-tumor activity, so you need not need additional in vivo studies. So this is what the question is. Next question is, should recovery be included, groups be included in the toxicology studies supporting first in human toxicology studies? So very important, when you see general toxicology studies like 28-day study, 3-month, 90-day study like that, in addition to the main groups, that is control, and the treatment groups like low dose, mid dose, and high dose, you have certain additional groups like TK group will be there, satellite group will be there, sentinel animals group will be there. So similarly, you have recovery group will be there. So what is the main purpose of the recovery group is, for example, if you have a 28 days repeat dose toxicology study in the case of rodents, that is in the case of rat, you have 14 days recovery period. So recovery period is the main purpose is that once the damage is done, if there are some adverse effects on the liver or kidney, is in the case of recovery group, where you will either select the high dose group like control, and the high dose group you will keep it in under recovery group. You will monitor the animal for next 14 days if it is a 28-day tox study, and you will not give any treatment in this 14-day duration. Whatever the main group animals will be there, they will be sacrificed on the day 20, 28 or 29, whatever the study is. Rest of for 14 days you leave the animals without treatment, and you will find is there any recovery is there. Okay, similarly if you have a 3-month study like 90-day study, you have 1 month recovery group in there. This is what the recovery group purpose in the case of general toxicology study. So now the question from the scientist is, how in the case of ICH-S9 guideline, the anti-cancer drugs which are used for advanced cancer treatment in the patient, do we need recovery group? Because whole purpose of ICH-S9 guideline is to reduce the timeline, to bring the drug more as fast as possible into the market, because you have to save the patient. Okay, so is there any necessary for the recovery? So let's see. So what is the answer which is given by the regulatory agency? The scientific assessment of the potential to recover should be provided. Here the guideline it is saying overall, if I understand it, is what is this saying, even though if you do not have any recovery group, or you are not waiting for the recovery data to have, at least you should be able to give either predictive recovery. So please read about this type of new terminologies with respect to recovery. Either it should show partial recovery, or predictive recovery, or potential recovery. So what is that meaning is this? For example, best example, most of these anti-cancer drugs, no, they will cause bone marrow depression or suppression, whatever it is. Okay, so once the treatment is stopped and the patient recovers, so we already know that bone marrow regenerates. So if it is, we need not wait for the 100% survival or recovery of the bone marrow. If it is showing some trend, temporal trend towards positivity, whether it is showing like 20%, 30% of bone marrow regeneration is taking place, it is showing potential recovery. Or if you understand the mechanism, we will take some other example like liver adaptation or whatever it is. If it is showing some trend, if it is showing some potential recovery, with that kind of data, if you understand the mechanism, with that suggesting that mechanism or adverse outcome pathway, you can still submit to the regulatory agency and you can defend yourselves. You need not wait for the like 1 month recovery like that. You need not wait. So once again we'll see. So a scientific assessment of the potential to recover should be provided in all general toxicology studies to support the clinical development. If you are starting phase 1 or phase 2, you have to submit this type of data to the regulatory agency like the FDA or Europe, Canada, etc. Although recovery group should not automatically be included. So it is saying that you need not include but show potential recovery in all general toxicology studies. Okay, what is the second statement? See, this information can be obtained by understanding that particular effect observed is generally reversible. For example, in the case of liver, if there is an adverse effect on the liver, we know that liver will regenerate. As we discussed here, bone marrow suppression also, or non-reversible by including a recovery group in at least one study. You are doing a series of general toxicology, at least in one. No need of including this recovery group in all type of repeat dose toxicology study. At least in one, if you are able to include and you are able to justify, that is enough. And one dose level to be justified by the sponsor. Okay. So please pause the video and read about what is partial recovery or predictive recovery or potential recovery. Okay. Next we will see question number 10. So question number 10 is also with respect to recovery only. So let's see what is this one. Whether we need to have recovery group in this case of for anti-cancer drugs who are used in the case of patients who are in the advanced treatment. So should recovery groups should be included in 3 month toxicology study to suppose phase 3. Now we are discussing about phase 3 trial. Okay, so recovery in the 3 month study is not specifically warranted. If you are doing a 3 month repeat dose toxicology study in the case of dog or monkey like that, do we need to have recovery group? No, recovery group in the case of 3 month study is not specifically warranted unless there is a concern by either understanding the mechanism or AOP pathway. If you are assuming, if you are able to anticipate or predict that yes, there is a cause for concern from the short-term toxicology study, like if you are doing a 28-day and you have find some kind of adverse effects which are have a potential concern, then at that time from clinical studies, or you have done phase 1, phase 2, in that you have some kind of adverse effects and you are assuming that it will definitely have some, that there is a necessity to have a recovery group to understand this adverse effect. At that time you have to include that recovery animals could address. So if you add a recovery group, if it is going to give you some additional data, that time only you have to add. For example, when a recovery group was not included in the short term, for example 28-day study have done, in that recovery group was not done and there was insufficient understanding whether a particular effect observed may be reversible or non-reversible. You have identified in the 28-day repeat dose toxicology study some adverse effect is there. Whether the adverse effect is treatment related, once the treatment is stopped, whether the effect will be reversible or new type of adverse effects you are able to see and unable to understand the mechanism or prediction, then at that time in the case of 3 month repeat dose toxicology study to support the phase 3 trial, you need to have at least in one of the general toxicology study, particularly in the case of 3 month, here in the phase 3 trial 3 month study, repeat dose toxicology study, you need to add recovery group. Another example is when the 3 month study are undertaken in the absence of clinical data. So till now there was not done it is not phase 3 but it is still phase 1, phase 2 has not been initiated, or with limited clinical data you have, but their sample population was less or you didn't do enough number of parameters. In that case also you need to have recovery group. A scientific assessment of the potential to recover from toxicity should be provided. As we discussed in the previous slide also, we need to understand the mechanism, the AOP pathway. Okay, for general toxicology studies used to support the clinical development, all the recovery groups should not automatically be included in all general toxicology studies. So always 3R principle is important. Whether adding recovery group to each and every repeat dose toxicology study in the battery of general toxicology, it is unnecessary increasing the animal number. It is not going to add any new data. So that is the reason they are asking us no need to add this recovery group to all battery of general toxicology studies. Okay, a more direct approach using appropriate model can be appropriate to address a specific safety question. Okay, if there is a potential concern, in order to address the main cause, that type of particular appropriate model, in that you can use this question. Okay, so as I told, please read about partial recovery, predictive recovery, and potential recovery. So at least two questions they are asked. Maybe in exam also, DABT also they might ask. We don't know. Okay. Next question we'll see. Question number 11. Patients with cancer are often given supportive care drugs. Example, antibiotics. This is also very important. So whenever you are treating a cancer patient, no, that particular cancer patient is susceptible to get some kind of infection, bacterial infection or viral infection. So you will be putting that patient under antibiotic therapy. Also, the patient will show some nausea type of effects. So you will be giving some anti what is it, emetic drugs you will be giving. So these are called supportive treatment, right? So for those supportive treatment, so some questions are related to that one. Patients with cancer are often given supportive care drugs. Here example they have given in the guideline is antibiotics. Is there a situation where adding supportive care drugs to toxicology studies are appropriate? So for these supportive care drugs, additional toxicology studies, that is the question. So what is the answer given by the regulatory agency? We'll see. Treating affected animals with appropriate supportive care during toxicology studies can be appropriate in some cases. What happens, whenever you're treating this anti-cancer drugs, no, the toxicology test in this healthy animals you are doing. So there is a thing that these animals, whether a rat, dog, or monkey, they become weak. So you have to rehabilitation. In the case of in addition to 3R principle, we have 4R principle. You have to take care of those animals. So example they have given, when secondary infection due to immunosuppression, this anti-cancer drug is there. It is used for tumor suppression, but in addition to that, it is also causing immune suppression to those animals, whether it is a dog or monkey like that. So in such cases, is observed on the study. So you need to treating affected animals with supportive care during toxicology studies can be appropriate in some cases. What are those some cases? In case of immune suppression is observed on the study. Okay, giving supportive care prophylactically before finding it to all animals is generally not recommended. Okay, nothing has happened. Before only you are giving the animal antibiotic. Before only you are giving antiemetic. That doesn't make any sense. Okay, so prophylactically giving supportive care to these animals is not recommended as per the ICH S9 guideline. Next question we'll see question number 12. Is there any guidance on the need for abuse liability studies for drug developed under ICH S9 guideline? So when I was reading about this, no, I was trying to understand what is this abuse liability. Generally, when we come across abuse liability, we'll see only with respect to abuse. Drugs of abuse will be recreational drugs like morphine or fentanyl. These type of drugs will be seeing. Then I try to connect it. Then I understood during this cancer treatment, patients know anti-cancer treatment to these patients know patients are under tremendous pain. So in order to reduce their pain, these doctors and the physicians, they will give pain relieving drugs. So in that they have morphine and fentanyl is also there. Okay, to reduce the to keep to comfort the patient, they will be given. But over a period of time, these patients might get addicted to this dependent type of drugs. So getting withdrawal from these type of drugs will be very challenging. So probably the question might be related to this one. So we'll see. First we'll understand what is abuse liability. I don't know much about this thing, so you also pause the video, go back and read. But whatever little information what we can discuss here, we'll see. First we'll understand what is abuse liability. Abuse liability or abuse potential is a pharmacological term defining the likelihood that a psychoactive substance, that is like morphine or those pain relieving will be there, no, will be self administered for non medical reasons. If the person is having cancer pain, it is okay, but here you are administering for non medical reasons leading to addiction, physical dependence, or other harmful consequences. So this is the meaning of go back to the question once again. Is there any guidance on the need for abuse liability studies for drugs developed under ICH S9 guideline? So what is the answer regulatory agency is giving is non-clinical studies for abuse liability are generally not warranted and not required as per ICH S9 guideline to support the clinical trials, whether it's phase 1, 2, 3. or either marketing of the pharmaceutical. Okay, even for the marketing also you need not have any abuse liability study for the treatment of patient with advanced cancer. Okay, so stop the video, read, understand in detail more. Next question is question number 13. Question number 13 is, what is the utility of cross reactivity studies for tissue cross reactivity studies for biopharmaceuticals? As I told you, most of these anti-cancer drugs are tyrosine kinase inhibitors, JAK kinase inhibitors, or ALK inhibitors. Some of the origin is not like traditional drugs, but these are aptamers or oligonucleotides or peptide proteins, antibodies. These are the nature of these molecules is like that. So these are biopharmaceuticals or biotechnology derived products. So is there tissue cross reactivity is important, whether any immune system is involved, sensitization is involved, that will be reading in this question. So what is the utility of tissue cross reactivity studies for biopharmaceuticals containing a complementary determining region, CDR, that is monoclonal antibodies, antibody drug conjugates, ADC, that fall under ICH-S9. The nature of this anti-cancer drug, new drug is either it is monoclonal antibody or ADC. Do these studies need to be conducted? Whether, since the nature of the investigational drug is antibody related is there, is tissue cross reactivity is needed or not? That is a question which is asked by the scientist. So what is the answer which is given by the regulatory agency is, in general, tissue cross reactivity studies have little utility. Okay, they are having not much significance and are not needed with initial first in human study. So FIH, if you are starting a phase 1 study at this particular point of time, this tissue cross reactivity is not needed. That is the thing which is explained by the guideline, or later in the development, unless there is a specific cause for concern. Again, it is a case by cases. If you find that there is a potentially threatening is there, such kind of issues are there, then only you have to think about this adding tissue cross reactivity study. In cases where there is no pharmacological relevant species, this is again very important. If you look into these molecules, whether it is ADC, that is antibody drug conjugate, or it is antibody related drugs, uh, you cannot do similar like that of general, like you cannot follow ICH M3 guideline. For this you have to specifically follow ICH S6 guideline, that is biotechnology derived products, because if you are doing as similar to that of general toxicology studies, and these, whether it is antibodies, they have a specific target. If the target is not there in that particular, you are doing a rodent model, you are doing a rat study, in that the target model is that receptor is only not there, how can this monoclonal antibody go and bind in the case of rat? So this is called pharmacologically irrelevant species. So you have to identify the species, whether it is rabbit or dog or monkey, in which the receptor should be there. That receptor should also be there in the human, and this monoclonal antibody should go and bind to that receptor. Then only it is will be considered as pharmacological relevant species. So in cases where there is no pharmacological relevant species, so there are challenges for these biotechnology companies, where they do not find any relevant species in that particular receptor. That particular target will not be there even in the case of monkey or in the case of dog also. So human tissue cross reactivity, so in that cases cell lines, human tissue cross reactivity or alternative method should be considered for the first in human studies. So it is much more challenging for those scientists to decide. For example, if it is a general molecule, based on whether it is dog study or it is monkey study or rat study, you will have a noel. Okay, by fractionation of that noel, by converting it to HED, you can decide the first in human dose. And in the case of anti-cancer drug also, there are certain places where you can have either STD10, that is severely toxic dose, that is STD10, or HNSTD, highly non-severely toxic dose, fractionation of this one. For example, in the case of STD10 by divided by 10, or in the case of HNSTD divided by 10, I guess. STD 10 by 100, that the uncertainty factor, correction factor is 100, whereas in the case of HNSTD by 10 you will be doing fractionation of HNSTD by 10 you will be doing, that will be considered a first in human dose. But in the case of those anti-cancer drugs, their origin is a monoclonal antibody or antigen drug, antibody drug conjugate, and for which there is no target only, there is no relevant species, no dog or dog monkey which is having this receptor. In such cases, getting a FIH, what is the safe dose that can be used in the case of first in human trials, is very challenging to the biotechnology scientists. So we'll discuss some questions on this in some other videos. So let's go to the next question. Question number 14. Question number 14 is, the guidance allows for the testing in only one species, as we are discussing now. So this section we are discussing only on non-clinical aspects only. First part we discussed mostly on the introduction, the scope. Now we are discussing what type of animal trials. Like we have discussed recovery groups, we have discussed about species also. Now one more question with respect to species only. So the guidance ICH S9 guideline allow for testing only in one species if there is a potential signal for embryo fetal lethality or teratogenicity study. So generally for these type of drugs, no, fertility studies, that is segment 1 as well as segment 3, that is prenatal postnatal studies, are not needed, because already these anti-cancer drugs, no, they have a potential threat that they might affect your male reproductive system or female reproductive system. Okay, once you are having advanced cancer also, you will not try to end up getting pregnant also. And similarly prenatal postnatal also, already rapidly dividing cells, this will definitely affect. PPND studies are also not. But coming to segment 2, EFD study, that is the embryo fetal developmental study, you need to do the study. And here the guideline is saying in one species. Generally EFD studies need to be done after the thalidomide tragedy incident, as per ISH M3 R2 guideline or ISH S5 guideline. If you see, which is specifically for reproductive developmental toxicology, it states that EFD studies need to be done at least in two species, that is one rodent and one non-rodent, that is rat and rabbit. This is mandatory. But in the case of ICH-S9 anti-cancer drugs, there is an exception. You can do it in only one species. So if there is a positive signal for embryo lethality or teratogenicity, one species is enough. If a clear evidence of embryo lethality or teratogenicity is observed in dose ranging findings, and these dose ranging findings can be even non-GLP also, in one species, is a definitive study in that species recommended? So this is the question asked by the scientist. What is the answer given by the regulatory agency? Will see a definitive study is generally not warranted. So if you have a dose range finding study and it is showing a positive signal, it is showing that yes, there is a possibility that this anti-cancer new drug can cause affect fetal as well as embryo, then no need of doing definitive study, no need of doing another GLP or main study. You can submit the non-GLP study, you can submit the dose ranging finding study to the regulatory agency, and they will accept it, and you can defend it by using ICH-S9 guideline. So a dose ranging, a definitive study is generally not needed, not warranted, if a dose range finding study including a non-GLP shows clear evidence of embryo fetal lethality or teratogenicity. This dose range finding study in a single species would be sufficient to support, including marketing, not only phase 1, 2, 3, 4 trials, phase 1, 2, 3 trials, as well as for marketing of this anti-cancer drug, can also be supported by using this non-GLP dose range finding study. No need of doing it in another rat, another rabbit, or like that. Okay. Next we will see question number 15. Section number 22.5 describes the use of alternative assessment for biopharmaceuticals. So as I told, most of this anti-cancer drug origin might be biopharmaceuticals. So use of alternative assessments, we'll see what is the answer which is given by the agency. Is there any role of alternative in vitro and in vivo assay for small molecules in reproductive toxicology assessment? Answer is yes. Alternative assessment may be used to aid the safety assessment of reproductive toxic risk. So instead of doing in vivo assay also, if you are able to support to the regulatory agency with the alternative, lot of alternative models are coming. Maybe it might be zebra fish model or some kind of cell lines through which you can able to address this reproductive risk. Then also at the time FDA or Europe agency will, regulatory agency will accept this data. Okay, alternative assay always in compliance with 3R principle it is. Okay, next question number 16 we'll see, when the only relevant species is a non-human primate. So in the case here it is monkey. The mechanism of action is expected to yield a reproductive toxicity risk or knock out the animals or kill the animals. Use of a surrogate biologics. Surrogate biologics means, and the actual whatever the investigation new drug, whatever the biologic chemical is there, no, that is not having any receptor in that particular model. But if it belongs to the same class of drugs like tyrosine kinase inhibitors or ALK inhibitors, like that same class of drug, means surrogate biologics in rodents have demonstrated a reproductive risk. Okay, how I should explain? For example, cephalosporins, cefexamine, cefadroxil, like that will be there. No, if one is able to prove in the rodent model, it was able to address the reproductive risk. No, should these approaches be considered sufficient for hazard identification? Should a study in non-pregnant NHP be conducted? Actually, we'll see what the answer is given by the agency. A weight of evidence assessment of reproductive risk should be provided. Very, very important. Same class of drug, surrogate biologic rodent, it is having in a rat, it is having receptor and it is shown reproductive risk. In the non-human primate, probably whether again you need to do this test or not, that an NHP study to assess the hazard in embryo fetal development should not be considered. Very, very important. In compliance with REAR principle, consider default approach if the weight of evidence clearly indicates a risk. An EFD study in NHP not warranted, because same class of drug, surrogate biologic as shown positive in the case of rat study. So no need of doing in with this investigational drug in the monkey or yeah. Development toxicity studies in NHPs can only provide hazard identification according to ICH S6. So as per ICH S6, which is go back and read about this biotechnology derived guidelines revision R1. So it only gives hazard identification only. It will give you the expected reproductive hazard should be appropriate indicate in the label. So whenever you have FDA label or Canada label or SMPC in the case of Europe, mention this kind of risk in the case of your label. You mention, even though we have not done EFD study, but same class of surrogate molecule, same class of drugs. For example, statins, they show the certain type of this thing, or anti-epileptic drugs, they have cleft palate, teratogenic risk will be there. No need of repeating in with the same class of molecules. Again, another new EFD study. Next question number 17 is there a need for non-clinical lactation and placental transfer studies? Because if you see in the FDA label also, whether the drug has been transported through through milk, through lactation, and it has caused, so these kind of things already we have discussed. PPND studies are needed or not. At this point, it is not needed. So there is no specific need for lactation or or placental transfer studies as per the guideline. Next, last slide we'll discussing question number 18. Question number 18 is which and how many in vitro genotoxic studies. So now we discussed about tissue cross reactivity, we discussed about recovery groups, we discussed about reproductive studies. Now we are coming to genotoxic studies. Which and how many in vitro genotoxic studies? First of all, you have to understand most of these anti-cancer drugs know their mechanism. One of the mechanism might be DNA damage. So these will be cytotoxic tests. So if these anti-cancer drugs you are doing some genotoxic test and if it will show positive, that doesn't stop the drug development. That you have to keep it in your mind. Okay, it is known effect. It is going to cause DNA damage. Okay, these anti-cancer drugs. Okay, so that doesn't stop for the drug from developing or go to the next stages of development, but you know the risk and the risk has to be mentioned in your label or it should be mentioned to the regulatory agency, whether it is FDA or EMA. Okay, so see what is the question: which and how many in vitro genotox studies would need to be positive in order to make in vivo genotoxic assay in vivo genotoxic assay unwarranted. So now there is a comparison between in vitro assay and in vivo assay. Whatever I spoke just now is in general. Okay, when the AIMS assay, that is bacterial mutation test, is positive, AIMS assay, the in vivo genotoxic test is not warranted. AIMS assay is positive, no need of doing in vivo test like micronucleases like that. When the bacterial mutation is negative, AIMS assay is negative, but in vitro chromosomal damage test, as chromosome aberration test or micronucleus test or mouse lymphoma TK assay is positive, in vivo genotoxic test should be considered. So that is the thing. Okay, so pause here, try to read how the genotoxicity associated with ICH-S9 guideline. Now, last question we'll be seeing, question number 9, which is related to photo safety. Okay, so section 2.9, photo safety testing states that if initial assessment of phototoxic potential based on physiological properties indicates a phototoxic risk. So based on whether PK properties will be there, the nature of the chemical, whether it is lipophilicity, how it is penetrating, IV, UV ray, like that, some physicochemical property test will be there, indicates a phototoxic risk. When should non-clinical photo safety studies can be conducted? Answer as per the agency is ICH S9 should be consulted for the timing of phototoxic studies. Means at what stage you need, whether at the time of marketing the drug or at the time of phase 2 or phase 3. So clearly go through the ICH S9 guideline. S10 should be consulted for assessment of photo safety studies. So coming to photo safety toxicology study, go back and read ICH S10 guideline, which is dedicated for for this photo safety studies. So read scope, the objective, at what timing you need, and what are the method, principle, all those things you read it. Okay, so thank you so much. Be curious. For any questions you can always write a mail to arya.priyanka18@gmail.com. So in this second we have discussed recovery groups, we have discussed about the importance of this photo safety, as well as we have discussed about the embryo fetal studies. Segment 1 to 3 studies are really needed or not, what kind of genotoxic studies are needed as for the ICH-S9 guideline. So mainly we have discussed what is the importance of non-clinical safety studies, their role, what can be waived off, what can be needed, what can be we have also discussed about relevant species, the nature of the anti-cancer drug, whether it is antigen drug candidate, bonded, or biopharmaceutical. So for those, how many studies can be really needed, or what can be, what are the alternate methods through which we can submit to the sponsor, and we can minimize the studies following the 3R principle. All the best for your exam.